And that's actually a cool example of coming up with a hypothesis, testing it, throwing it away.
Yeah.
Coming up with another one.
Mm-hmm.
It's called science, David.
Oh, yeah, I've heard of that. Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klonowski, your host for today's episode. Viruses lack much of the machinery required for replication and need to infect cells of the host to multiply. Can this need to gain entry into a host cell be their Achilles heel?
Our guest today is Dr. Melinda Brindley, Associate Professor and Director of Graduate Studies in the Department of Infectious Diseases. Melinda, it's great to have you here.
Thank you for having me.
So our go-to question to kick things off is to ask folks about their pathway to science. Were you one of the ones that knew you were going into science from kindergarten, or?
Yes, probably. I grew up with two pharmacists as my parents, and science, or drugs at least, were talked about on a normal basis at the dinner table. I always knew I wanted to go into science. Originally in high school, I thought I wanted to do gene therapy, or not necessarily gene therapy, like genetic engineering of plants. But then I went to undergrad and realized that experiments in plants take really long time, and I don't have patience for that. So I discovered that viruses are super fast, and you can do an experiment in a couple days. And so I focused more on doing experiments in virology.
That's better than redwood genetics, I guess.
Yeah.
Why plants? Did you just like the outdoors, or?
We always had National Geographic at the time, and golden rice had just come out, and they were, making vitamin A part of the rice genome, and that was gonna, and I just was like, oh, this is really cool. I wanted to be in that field, I guess, at the time. But yeah, it's just really slow to do those experiments.
Right. So now that you're working on viruses, what is the main question that you address in your research?
So when we started the lab, we always focused on how viruses get into cells. And originally we were focusing on the proteins involved, protein-protein interactions. But one of the things that we've been focusing on is how some viruses can get into the cell not through a protein-protein interaction, but through a lipid-protein interaction. So cells pick up things on a normal basis, like dying cells, through an interaction with phosphatidylserine, which is a unique lipid that's not normally on the outside of cells, but during apoptosis, when they're dying, they switch this lipid to the outside. And then cells, I think there's like 24 different PS receptors that cells can produce. And if the cells pick up or notice PS on the outside, they'll just pick up whatever that is and bring it inside the cell and try to degrade it and basically get rid of it. But some viruses utilize that pathway to enter cells. And so rather than a protein on the surface of the virus interacting with the cells, it's actually the lipid on the viral particle that's interacting with phosphatidylserine receptors on the cells. And so the cells basically are picking up viruses because they think it's just apoptotic debris. Then the virus gets in, it realizes that it's inside the cell and can escape and infect the cell. And so it's called apoptotic mimicry for viruses. So basically the viruses are dressing up like an apoptotic cell. So we've been looking at how viruses can incorporate PS and increase the level of PS on the viral particle in order to infect the cell. And then we've kind of expanded that and just really started to look at how lipids in general change during viral infection. Like most people, they'll see a textbook or something and the plasma membrane would be just kind of a line on the cell. But actually, like, there's 100 or tens of thousands of different lipids within a cell. And specific ones are needed for different regions. And so we have a collaborator with chemistry that has the ability to identify hundreds of thousands of different types of lipids. And so we can infect the cells, take infected cells or uninfected cells and give them to her, and she will tell us which lipids are changing. And then we try to figure out if those lipids are changing because the virus needs those lipids, or is the cell trying to prevent the virus from replicating? So it's altering the lipid environment.
I kind of want to take a step back a ways and talk about, in general, how viruses invade a host cell. Do any of them have an active process, or do they just induce the host cell, trick the host cell into taking them up?
So viruses are unable to do anything outside of a host cell. So they're just going to be floating around. And if the virus binds to or interacts with the surface of the cell in a particular manner, then the cell will uptake that virus. So some viruses have glycoproteins on the surface that will interact with particular receptors on the cells in a very lock and key like specific interaction, like HIV interacts with CD4 or influenza virus interacts with sialic acids. Those are very specific interactions.
Or SARS-CoV-2 with the ACE.
Mm-hmm.
Right?
Yep. And so just for a number of viruses that we're studying in the lab, they can infect virtually all kinds of cells. And they were trying to figure out, well, how could it be so universal? And that's where it's more these PS receptors on the cell surface are not interacting with a particular protein on the virus, but just a lipid. And because those lipids are stolen from cells to begin with, they're the same lipids that are on apoptotic cells.
Yeah, so I want to get to that because you mentioned that viruses can't do anything outside of the host cell. So the lipids that they are using essentially are our lipids.
Yes.
Right? Because the virus buds. Do you want to just talk real quickly about the budding? So
viruses can't do any kind of metabolic things outside the cell, but in an infected cell, at the end, if it's an envelope virus, meaning a virus that has some kind of lipid membrane, they're going to have to steal that lipid from that infected cell. And so some viruses will do it in like the ER membrane, like Zika virus or dengue virus steals the ER membrane. But most of the viruses that we study in our lab bud at the plasma membrane. So right at the end of their life cycle, they're going to just pick up a little piece of that outer plasma membrane. And usually there's some glycoproteins in there too. But all of the lipids were present in that infected cell.
So there's really 10,000 different lipids?
Mm-hmm.
Holy cow.
Yeah, I don't know many of them, but like over in chemistry, they can. So part of it is, right, there's two fatty acid chains. They can be saturated or unsaturated. If they have some double bonds, those double bonds can be in various locations within the lipid chains. And so if you start thinking about how many carbons are present and where the double bond could be, the number of different types of lipids start to increase dramatically.
So what viruses use the lipids?
So any envelope virus is going to need lipids at some point in their virus life cycle because they're covered in the lipid envelope. And so we study, for the most part, negative sense RNA viruses, and those aren't known to interact or replicate on membranes. But positive sense RNA viruses like SARS-CoV-2 or dengue, those actually replicate their genomes on membranes, and they do that to hide from the host. So basically, innate immune sensors would notice that there's double-stranded RNAs if they were just doing this anywhere in the cytoplasm. And so they make these little pockets of lipids to hide from the host cell. So they're stealing the host, and they really dramatically change the lipid environment in the cell during that process. And if you look under EM, you can see all these little, they'll call replication machineries or spherules, depending on which virus that you're talking about. But the negative sense don't do that. They have a protein that's coding their genome, so they don't make these double-stranded RNAs in the process, so they don't need to replicate on lipids. But because of that, no one really studied extensively how the lipid environment changes when negative sense RNA viruses replicate, because for the positive sense, you can like visually see the lipids. So many people had been monitoring how that's changing. So we infected cells with vesicular stomatitis virus, and we looked to see what changed, and we see thousands of things change during virus infection. We didn't really follow up on figuring out which changes are important or not for that virus. But then we repeated the experiment with lymphocytic choriomeningitis virus, LCMV, which is an arenavirus similar to Lassa that you can actually use in the BSL-2. So we infected the cells with a chimeric LCMV. So it's 4 proteins are present in that virus: the polymerase protein, the Z protein, the nuclear protein, and then the glycoprotein. And so we have a chimeric of 3 proteins are all LCMV, and then the glycoprotein's been replaced with the Lassa glycoprotein. So the entry pathway looks like Lassa.
So this is a way to work and kind of study Lassa, but under BSL-2 conditions, right?
And maybe you want to give us a little background on Lassa and why it's so dangerous. And if you were working with the holo Lassa virus, how would you be working with it?
Right. So Lassa is a BSL-4 pathogen. So that's a pathogen that can cause severe disease and pathogenicity that requires basically one of those like astronaut outfits to work in. So you have to completely be contained in a suit that is full of air pressure, and you're working in a BSL-4 where there's never going to be an opportunity for you to be exposed to the virus. So Lassa is an arenavirus. It's transmitted from rats to people, usually through either aerosolization or like interaction in the skin. The rat feces is going to be full of virus, and if you're cleaning up your house and you have scratches on you, you could like get infected through that, or you can get infected just through like sweeping and breathing in a lot of the. So for the most part, it's interesting. Most people don't appear to get severely sick from Lassa for the most part. So most cases are actually asymptomatic, but the few that do get symptomatic, they can cause severe disease and hemorrhagic fever. And so if you are symptomatic, about 20% of the people will end up in the hospital, and some people won't survive infection, but it's thought to only have about like 1 to 5% mortality rate if you actually look to see how many people are actually have antibodies to Lassa, that it was clear that they've been exposed at some point, but they don't remember ever being sick.
Right.
But it is especially dangerous in pregnant women. So pregnant women who are exposed to Lassa, not only will they lose the baby, that frequently the mother will actually pass away too, because it's very severe disease.
So you're taking just one gene from Lassa and putting it in vesicular stomatitis virus.
So we do that, but we also have this chimera that's all arenavirus. So LCMV is this BSL-2 arenavirus, and we'll just move the glycoprotein into that. So this particle looks like the arenavirus particles do, whereas the VSV particles look very different, and they actually replicate slightly differently. And so they're slightly like a step away. But we use those particles in addition to the LCMV Lassa chimeras. But so yeah, we infected the cells with this virus and then looked to see what changes happened in the lipidome. And one of the biggest things that we saw is we saw a huge increase in triglycerides. And this is just in cell culture. So triglycerides are little fats that have like 3 fatty acid chains. Like people usually know about them because if you have high triglycerides in your blood, that's not a good sign for heart disease and whatnot. But what we could do in the lab is we could say, okay, well, what if we use one of those drugs that people would be on to stop triglycerides?
Statins?
Yes, a statin, to see if that makes the virus stop replicating. Because we didn't know if the triglycerides were super high because the virus needed those triglycerides, or because the cell was trying to stop that virus from growing. And so when we did that, we didn't see any effect. So even though we have these huge upregulation, like 10 times more triglycerides in the infected cells, there was no effect on the virus. And so we thought that there would be some effect in one way or the other. But then we decided to look at those lipid pathways a little bit more carefully. And so triglycerides are usually made if the cell has too many fatty acids. So fatty acids are toxic to cells. And to prevent that toxicity, the cells will package them in diacylglycerol and triacylglycerols, and then put them in little lipid droplets within the cell. And so if we went upstream of triglyceride synthesis and stopped fatty acid synthesis by, again, using some drugs that people are taking for various lipid regulatory issues, then we saw an effect. So if we stop fatty acid synthesis, then the virus isn't able to replicate. So Joseph in the lab, one of my students, is looking to see what exactly those fatty acids are needed for in the virus. So 2 of the 4 viral proteins actually get modified and have a fatty acid added to them. They're myristylated, so they have the C14 lipid added to them. And we thought that that would be like a logical reason why fatty acid synthesis would be regulated or required for these viruses to replicate. But interestingly, if we take the cells that have been blocked for fatty acid synthesis and give them back that C14 lipid, the virus wasn't rescued. But if we give them back this longer lipid, everything went back to normal. And so we actually don't think it's blocking the fatty acids is messing up these normal myristylation processes, but we think it's some other phospholipid that is requiring this longer fatty acid chain that's required for the virus to replicate. But we still have a handful more experiments to figure out exactly which lipid is required.
And that's actually a cool example of coming up with a hypothesis, testing it, throwing it away.
Yeah.
Coming up with another one.
It's called science, David.
Oh, yeah, I've heard of that.
So we talked, I'm going to go back to the phosphatidylserine, which you mentioned that you're finding these viruses to be coded in. And you alluded to this when you started talking about they're normally expressed on apoptotic cells, right? So that's just a way for the virus to get in, right? And it's not going into a degradation pathway. It's just, once it gets in, it's just letting loose, right? So it's kind of the sheep in wolf's clothing. Fascinating.
Yeah. No, so the cell will normally pick up that material and take it into the endosome. And then inside that endosome, they'll start pumping protons across, making it more acidic. And that'll activate a whole bunch of proteases that will start cutting things up and normally degrading it. But that acid and or those proteases are actually triggers for the virus to escape that endosome. So some of the viruses utilize that low pH, and that low pH causes these dramatic conformational changes in the little machines that the viruses have on the outside to fuse with the cell and escape. And then Ebola virus actually uses one of those proteases that are activated during low pH. And so the protease then cuts the glycoprotein up, and only after that cleavage event can the virus actually see its receptor and cause the fusion event to happen. So it's their way of knowing they're in the right time and place to infect the cell.
So small and so devious.
Yes.
So I read probably on your lab website that a virus like chikungunya, which, yes, that's the real name of the virus, it has multiple routes of invasion.
Yeah.
And that's not something I think of for a virus. I think of it as having just, I guess my model is the lock and key sort of thing. And it seems amazing that with a minimal genome, it can have that kind of variety of invasion tools.
Yeah, we've been noticing that for several viruses, and it's impressive that they have the ability to just basically interact with whatever is given to them. So chikungunya virus is a virus that needs to infect both humans and the mosquito host. And so because those are so divergent hosts, maybe that's part of the reasons why it has, utilizes anything that it has to get into the cell. But there's definitely protein receptors for chikungunya that are lock and key, like MIXRA8, and chikungunya virus has this nice protein-protein interface that allows it to get into some cells. But you can take a mouse and knock that protein out, and the mouse still gets infected with chikungunya. It doesn't get to quite as high titers, and then they won't see, chikungunya usually causes like severe joint pain and swelling, and they won't get swelling of the joints if they knock out the MIXRA, but the titers are still plenty high, and you can actually still transmit to mosquitoes. So it's clearly not the only receptor. So that lock and key one that we know of is thought to be much more important for pathogenesis, but chick can also use a lot of kind of more generic ways to get in. So it can also interact with just heparins, which are kind of like sugars on the outside of cells, and it can get in through that way, and it can also get in through the PS receptors. So it's just depending on what the cell has, it will use whatever it is. And we really want to know what it uses to get into mosquito cells. And so although there's been a handful of suggested receptors, none of them have really been proven to be definitive. And so what we decided when a previous student tried to say, well, we know how to block infection in viro cells using PS receptors, or HAP cells require the heparin sulfate, the sugars, and then the MIXRA. And so we threw all of those things that would normally block infection in different mammalian cells, but nothing blocked it in the mosquito cells. So it looks like they can get in through some other pathway. So there are definitely other people looking at that more specifically, because the mosquito is kind of trickier to work with. Like you can't just use a normal Cas9 CRISPR library to look in the mosquito. So people are developing new tools so that you can do those same kind of experiments that you would do in mammalian cells in the mosquito host.
Yeah, I want to point out that, sometimes my colleagues mock the viruses and some of them having a very small genome, but they can do a lot, right, with that small genome. They've adapted ways of using us mostly, really. So Melinda, often the headlines are stolen by pathogens that don't contribute to much morbidity or mortality. I'm talking to you about Ebola, but there's common viruses, right, that have a greater public health and economic burden. In terms of both death and disease, what viruses do you think are the biggest threats?
Right now in the United States, it hurts my heart to hear that measles is in the headlines so often right now. So I did my postdoc on measles. So I spent 5 years of my life focusing on measles. about 15 years ago now. And I really thought moving and starting my own lab, like measles is not something that we should focus on because we have such an awesome vaccine. There's no reason to continue to work on something like this. But unfortunately, that's people and misinformation has been spreading and allowing that virus to come back into the headlines. And so we had the first death in the United States from measles a few months ago in decades. And then just last week or 2 weeks ago, we had a second death from SSPE, which is a complication from measles that can occur about a decade after you get the measles. And basically that virus was able to enter the brain and cause devastating issues inside the brain, and it's eventually lethal. It's so hard to hear.
That was one of the great public health victories.
Mm-hmm.
Measles, to me, is really interesting for a couple of reasons. One is that it was so common, and I think sometimes familiarity, it doesn't in this case breed contempt, but it allows you to think, well, yeah, it's just a common thing, but there's a lot of sequelae with it. And
yeah, measles is the most contagious viruses of people that we know of. It's so contagious that in order for that measles to survive in a population, you have to have a population of at least 500,000 people, because if you don't have that many people, not enough new people will get produced on a routine basis that can be reinfected to keep that virus alive. Because this is one of a virus that does not have an animal vector or animal host. Mm-hmm. So it's a purely human virus. We can eradicate this pathogen, similar to smallpox.
Right.
And we have an awesome vaccine that was developed in the late 60s and was really pushed through, and most people were getting the vaccine, at least in the United States, which is good because you need to have at least 95% of the population be immune to measles to prevent outbreaks because it's so contagious. So if you have a virus that is not quite as contagious, then you don't have to have as high of a vaccinated population or immune population to stop outbreaks from happening. But it really is quite high for measles. And that was successful for such a long time, but we had so much success that people forgot what measles can cause. So a lot of people think, oh, measles is just a itchy rash or something like that. But actually 1 in 10 people that end up getting measles will have secondary infections. They'll have an ear infection or pneumonia. Pneumonia is one of the highest rates of death from measles because they get severe pneumonia or they'll get encephalitis. The most recent outbreak that we're having right now is highest in decades in the United States, and 20% of the kids are ending up in the hospital. So it's not like this is an average cold. Everybody who has the measles will get a fever. It's very different than, Lassa we talked about, there's a lot of asymptomatic infections. People will get antibodies to it and we never remember getting infected. Measles, you'll know that you're infected with.
Right.
And then not only that, is it infects your B and T cells and kills them, the actual clinical isolates do. And because it's killing your immune system, then you don't have the ability to fight every other normal infection. And so it can cause something what has been termed immune amnesia, because it's going to kill some of your memory cells that had been developed to infect all their pathogens you'd already fought off. So now you're going to have to potentially do that again. So you get actually immunosuppressed for several months after you recover from the measles.
Absolutely devious.
Very devious. So when we're talking about these infections, right, and a lot of times you want to talk about this in terms of mortality, but you've also already alluded to the fact that this causes a lot of hospitalizations, right? Mm-hmm. And we're at a point in time where all hospitals are closing, where we're having shortages of availability of maybe rooms with people going into the emergency rooms with lack of healthcare coverage, and then parents, of course, that have to take off from work. So this is not just, a disease that we have to worry about in terms of mortality, right? Yep. Which is obviously worst case scenario, but we have other effects from this infection.
Yeah, and right now, at least traditionally in the United States, if you had a measles outbreak, every single person was traced because it's so contagious. They want to know who are the potential people that it could be interacting with and have the potential. So in public health, you have to decide when you're going to recommend vaccines. And so in order to make this as straightforward a process as possible, as also protecting as many people, we recommend the MMR vaccine at 1 year of age. But that's because about 85% of people will no longer have maternal antibodies that would prevent that new baby from reacting to the vaccine. Unfortunately, it's not 100%, right? So Some people will lose their maternal antibodies earlier than others. And so a lot of kids between 9 months and a year actually potential to get the measles if there's active outbreaks because they aren't immune anymore. They've lost their mother's antibodies, but they aren't of the age to get their new vaccine yet, or like the actual vaccine yet. And then even at 1 year of age, 15% will still have enough maternal antibodies that they won't react themselves. And so that's why we give a second dose right before you go to kindergarten. You'll get a second dose of MMR to make sure everybody is actually protected. And as long as we don't have ongoing measles infections walking around, this works great. And that's what it's worked for a long time. But now there's some cases of kids getting, measles at 11 months and they're in a family that would have vaccinated normally, but because they're not in a herd that is protected, they're getting exposed and potentially infected.
So what kind of vaccine is the measles part of MMR?
Yeah, so measles, mumps, and rubella is all attenuated vaccines. So all three of them are actually just weakened viruses, but you're actually getting infected with a virus that can no longer cause disease. So the measles virus, you get about 1,000 virus particles in the MMR. Measles is normally a respiratory pathogen that would infect the lung cells or alveolar macrophages early in infection, but when you put it in the arm, the vaccine strain, which was passaged and developed in eggs, right? Like, so they passaged it in chicken embryo fibroblasts for many, many, many generations. That vaccine now has an expanded tropism. And so a lot of people think about expanded tropism as it's going to cause more pathology or more danger. But this virus actually, instead of only infecting your B and T cells, it now has the ability to affect like almost every cell in your body. It uses CD46. And so it's going to just infect your skin cells, but it doesn't cause the same kind of disease in your skin cells. It gives your immune system plenty of immunogens to make good antibodies to, but it doesn't cause significant disease. It can cause fever occasionally. And so the rare cases of vaccine-associated issues with the MMR is usually from fevers, because some kids, when they're really young, they'll have seizures if they get fevers. And so that's usually one of the only known severe side effects from the MMR vaccine, and it's very rare. It's like 1 to 3 cases out of every 10,000 doses.
Since we're talking about vaccines, one thing maybe we could touch on is the advantages of an attenuated vaccine versus what we call a subunit vaccine, where you're getting just like one protein from the pathogen. So take it away.
So an attenuated vaccine is nice in that you're getting basically all the different viral proteins present, and your immune system will be able to make antibodies against all of those different proteins. The other thing is the virus self-doses or self-amplifies within the host. And so even though you're only getting 1,000 measles virus particles, those particles will able to replicate a handful of times and give the body additional material to make antibodies to. So you don't have to get multiple doses. Like the SARS-CoV-2 vaccines were just giving you one protein that you had to produce. And so you had to get a second dose those original times you were getting vaccinated. So usually you can use much less virus, much less dosage of particles for an attenuated virus vaccine. But there are some disadvantages. So if you're immunocompromised already, you shouldn't get an attenuated virus vaccine because something is wrong with your immune system. It is a replicating vaccine. You want to ensure that you have the ability to react to that virus appropriately. And so that's part of the reason why you have that herd immunity. So as long as other people in the community are getting vaccinated, then those few people that have severe immunocompromised problems don't have to worry about getting infected.
And because you're spreading out the immunogens across many different targets, you don't have to worry like you do about flu or COVID that are those subunits or targeting only one thing, because that one thing could mutate, right? You
somewhat
kind of expanded.
Measles is also very different from influenza and SARS in that the main place that you're going to make antibodies that actually cause the virus to neutralize happens to be really the only face of that protein that doesn't have any ability to drift. So you can't really get antibodies against another location, or measles virus has 2 different receptors that it interacts with. They're both on this like very open protein face within the protein. They're slightly overlapping. If you get any mutations within that region, usually the virus is less fit and it can't replicate, so you can't escape around it. But that protein face is completely open, and like our immune system just loves it and makes really good antibodies to it. So unlike influenza that is just really good at changing its outer coat so that the virus itself can still replicate, but the immune system doesn't recognize it anymore. Measles doesn't have the ability to do that. People in the lab have tried to like make measles virus evolve away from being able to be neutralized from the vaccine. It just doesn't work, which is great.
Oh, that's good news.
Yeah. So like this is a virus that we developed a vaccine back in the 70s. We're using the same vaccine now, and it's still as effective today as it was decades ago.
And we should say as safe as it's ever been.
Yes.
Correct. Melinda, so with the recent changes we're hearing about in the news from the CDC regarding vaccine schedules, is the MMR vaccine still recommended?
So the new CDC recommendations suggested that the MMRV vaccine, which is the measles, mumps, rubella, and varicella or chickenpox particular vaccine, is not used, but the MMR is still being recommended. So MMR, measles, mumps, and rubella, is still being recommended for children, both at 1 year of age and then again between 4 and 5.
I have read reports from that vaccine advisory committee, and it is concerning to say the least.
They're just trying to put a little distrust in the system, because if they do stuff like they just did and suggest that the MMRV vaccine is not good, the media does pick it up incorrectly and people read it incorrectly and don't realize that there's very nuanced things that they're changing, and they'll just see, oh, we shouldn't get the MMR vaccine anymore.
So moving away from measles and chikungunya and Lassa and Ebola and other things, what do you do in your free time?
Well, I have 3 kids, so they keep me a little busy. But personally, my husband and I are really good or into Legos. We do Legos a lot at night.
I've seen the Lego model of your lab in your office.
Yes,
that was
I'm
pretty
geeky
cool.
enough that we reproduced the lab scale model. But we also do all of the science, so we have all the NASA sets, and then we also do the modular sets. So any of the each year they come out with one new set, so we get that, and we'll do that in the evening. I like TV, so we usually watch do Legos and TV.
Are all flat surfaces in your home
Yes.
filled with Lego?
Yes. I mean, I've had to bring them into the lab because every set.
You probably
we have a Lego room now.
Oh, you do? Oh, that's fascinating. That could be a great field trip.
Oh, yeah. Well, with that, our guest today is Dr. Melinda Brindley, Associate Professor and Director of Graduate Studies in the Department of Infectious Diseases. Melinda, it's been great having you on.
Thank you.
Links to additional information about Dr. Brindley's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast. And if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nebitz, for research, editing, scheduling, and keeping us on track.